Alex will do the introductions this time. Alex, are you there?
Yes, I'm with you. Thank you, Morten. Very happy to be with you all, and welcome to the second day of the 8th ARDD, the largest conference in aging research in the biopharmaceutical industry. It is my great honor to introduce Professor Manuel Serrano from RB Barcelona in Spain. So he requires no introduction, and I would rather give him the more minutes to talk about his work in vivo rejuvenation using the Yamanaka factors. Only few highlight points that I wanted to make is that he published his in vivo rejuvenation papers and theory, I think, before the epigenetic aging clocks were published. So that was pretty pioneering work. And nowadays, we know that we can track aging with methylation and with many other sources of data. And it's very impressive that that work in vivo rejuvenation has been done even before those clocks emerged. He is also one of the key authors on a very famous paper that is probably the most famous paper in our industry right now, published in Cell, The Hallmarks of Aging, published together with Linda Partridge and Blasco and Krummer. So his work specializes, is focused on cellular senescence and cellular plasticity, cellular reprogramming. And it's great pleasure and honor to have him at the conference today. So Dr. Serrano, please, the floor is yours.
Thank you, Alex. Thank you very much for this nice introduction. Thank you, the organizers, for this opportunity, and to the audience for being there. As Alex was mentioning, I'm going to talk about in vivo rejuvenation using the Yamanaka factors. And I will mention the work that we are doing with methylation and epigenetics. So Alex anticipated this very well. I hope you see now my screen.
We can see it, yes.
Yeah, okay, in vivo rejuvenation. Okay, a few words about reprogramming. This is a cartoon that summarizes the current view about reprogramming in vitro. As you know, these 4 transcription factors are able to change the identity of the cells and make them embryonic stem cells, or iPS. But this journey doesn't happen smoothly. It is very tortuous and complex. And this is what is represented here by different stages. There is initially a dedifferentiation and what we call, or some people call, partial reprogramming. At this point, the cells, rather than having a new identity, basically they don't have identity. And it is very heterogeneous, and this is what is represented here with this funnel. There is a high heterogeneity, but if the Yamanaka factors are maintained, there is a point of no return, and then the cells will acquire the new identity. In vivo, something very similar happens, and this is work that we did with Maria Abad, already published a number of years ago. We were able to dissect the same stages, a progressive dedifferentiation in many tissues. This is induction of the Yamanaka factors using doxycycline and a ubiquitous promoter, ROSA26. In this case, after the point of no return, what happens is that there is the risk of teratoma. Because if iPS are generated in vivo, iPS generate teratoma. So this is the demonstration that full pluripotency is achieved in vivo in many different tissues. Okay, so once we know that this is possible, let's go now to the concept of rejuvenation. And the rejuvenation is focused on this partial reprogramming without the acquisition of the new identity. It's partial reprogramming and then recovery of the parental identity. There is no change in cell identity. There is a partial change, and then recovery of the original identity. Because in the rejuvenation field, in theory, we don't want to change the identity of the cells. We want to rejuvenate the cells without losing their identity. This is an example of what happens in the pancreas. The exocrine pancreas is the one that shows more reprogramming and earlier evidence of reprogramming in our mouse model. This is an island of acinar cells that look normal in the histology. This is after 1 week of activating the Yamanaka factors in vivo. But look at the rest of the pancreas. The rest of the pancreas is dysplastic. We don't have a better word for that. And is full of cells with different morphologies. As you can see, it's difficult to find a pattern. It is very transformed, and we are now, we have a working very well in advance, doing single cell RNA sequencing of these cells to understand what kind of new identities or new of cellular identities are here, because here the cells have lost their differentiation state. If the Yamanaka factors are continue expressed, these cells will give rise to IPS. But at this point, they are not IPS. And actually, this process here at this point is reversible. And this is what is represented here. So this is the slide I showed before. And now what we are going to do is we are going to have the Yamanaka factors on for 1 week. And then after a week, we switch them off. And what happens is that this loss of differentiation, this partial reprogramming, is reversible, as I was anticipating before, and the cells reacquire the same identity that they had. So there is a period of recovery, and after that period of recovery, 1 week or 2 weeks, what we observe is that the pancreas now is absolutely normal, histologically normal. When we do this process, we don't observe in the long-term teratomas because the cells don't have time to acquire pluripotency. And now the question is whether this process of partial reprogramming and then recovery has rejuvenated these mice. I have to mention here the pioneer work of Professor Belmonte in the Salk Institute and Sebastiano in Stanford. They reported that multiple cycles of the Yamanaka factors, short cycles of 3 days or 1 week, are able to rejuvenate cells in vitro, human and mouse, and they are able to extend the longevity of progenic mice. We have been working on this along the same lines, but instead of doing multiple cycles, what we have done is just a single cycle. We want to study a single cycle because we think that in this way we can dissect the mechanism better. Instead of the accumulation of many cycles, we are analyzing just one. And the other thing that is different is that we are using naturally aged mice, mice that are either 1 year old or 2 year old. I will go quickly into the analysis. We have done a number of omic analyses, and we started with the transcriptome. I'm showing you here data in the pancreas, although we have similar data, maybe not as nice, because as I said, this reprogramming is more broad and more synchronized in the exocrine pancreas, but it also can be detected in other tissues. We did the RNA-seq of the mice. These are 4 mice, and here there are 5 mice that are young or old, and we selected those transcripts that change with aging. So in a way, we are constructing a clock. Because we are selecting those that change with aging, of course, principal component analysis nicely separates the two populations, the old and the young, because this is a condition. Now the question is, where the mice that were exposed to the Yamanaka factors for 1 week, where are they located? And they are more or less in the middle. And we can ask which transcriptional programs have been rejuvenated in these old mice after the Yamanaka factors. And the one that appears in the top, the most prominent one, happens to be a famous and very old friend in the aging field, that is mTOR signaling. mTOR signaling, here are the different genes of this signature. These genes are upregulated in the old mice compared to the young mice. Okay, when we reprogram the old mice, now these reprogrammed mice, they behave as the young mice. So this upregulation of the mTOR signature is reduced after 1 week of the Yamanaka factors in old mice that are 1 year old. We have done a similar work in collaboration with Wolf-Rayk, but in this case, analyzing the methylome. In the same way, the laboratory of Wolf-Rayk at the Babraham, they generated a clock, if I may say that, by using reduced representation by sulfate sequencing, and they selected those few regions in the genome that change methylation with aging. And they separate the old from the young in the pancreas. And the question is where these reprogrammed mice are. And again, they are in the middle. So this one cycle, remember, this is only one cycle. This one cycle is able to rejuvenate some of the changes that have happened with aging in the methylome. We are taking advantage of this to try to get some mechanistic insights. We have distinguished which genomic regions acquire methylation with aging and lose methylation with the Yamanaka factors, or the reverse, which regions lose methylation and then reacquire the methylation with aging. And we are distinguishing promoters and enhancers. And I'm going to go to the mechanistic insights that are more robust, let me say, put it that way. Similar results have been obtained in the liver and in the spleen. So this is a summary of, I want to mention 3 points here. The first one, this is about the changes in methylation. Please remember that by large, 99% of the CPGs don't change their methylation with aging. We are focusing on those few changes, is a small percentage that change with aging. Okay, first message is that the majority, we have compared the changes in methylation immediately during the expression of the Yamanaka factors. So we express the Yamanaka factors for 1 week and we analyze what is happening there that 1 week. And we also analyze what happens after 1 week of recovery and after 2 weeks of recovery and after 4 weeks of recovery. And we found, to our surprise, that about half of the changes are unstable. Half of the changes that happen immediately after the Yamanaka factors, they disappear with the weeks, with the following weeks. They are not stable changes in the methylation. So I think that this is one lesson, and is that when we measure the methylation clock, we should wait for the stable changes or changes that are maintained after a few weeks. The other thing that we can ask is when these changes are acquired. We were thinking that all the changes are happening just because the Yamanaka factors are expressed. And this is the case with the remethylation. These are the changes that methylation sites that are lost with aging, and then they are remethylated with OSKM. So OSKM remethylates these sites. Some of them are unstable, but the ones that are persistent, they are acquired at the time of expression. The surprise is the demethylation. We found that almost very, very little demethylation happens during the expression of the Yamanaka factors. Most of the demethylation that is rejuvenating happens after the Yamanaka factors. We interpret that this is part of the remodeling of the chromatin, that the Yamanaka factors change the chromatin, and then there is a reconfiguration to acquire the original parental identity. And in this recovery of the original identity is when this demethylation happens. So here we see a different behavior between methylation, gaining of methylation, and losing methylation. And we think that this may have mechanistic interpretations that we are trying to understand. Let me show you here an example. Here we are focusing on a single CPG. This has been measured by pyrosequencing by our collaborator, Mario Fraga. And so this would be the amount of methylation in one single CPG in young mice and in mice that are 2-year-old. You can see that there is an increase in methylation. Of course, it's heterogeneous. This is aging. This is very heterogeneous. But these are mice that are 2-year-old. This is indicated here, 2-year-old. And then they have been reprogrammed with the Yamanaka factors for 1 week and recover for 2 weeks. After 2 weeks of recovery, we see some demethylation, but not a lot. You can see also here in this another side. But after 4 weeks is when we see the full loss of methylation in these particular CPGs. So this is an example of how this demethylation happens post-OSKM. And with this, I'm going to conclude this work with the message that a single cycle of the Yamanaka factors in naturally aged mice is able to rejuvenate molecular features of aging. Molecular features in the epigenome, in the transcriptome, and also in the metabolome. I'm going to go quickly here. This is work that is published in stem cells reports. We have used cycles of reprogramming as well to try to see if we can see rejuvenation in the brain, particularly in the de novo neurogenesis in the dentate gyrus. This is very associated, is very well associated to the capacity of the brain to memorize. And this is an example of one of these tests. I'm going to go quickly here, sorry, because of time. When mice are exposed to 2 objects, and then if a few hours later are exposed to 2 objects, one of them is the same and the one here is new, they spend more time, 75% of the time, in the new one. They remember that this one is an old friend. And this is the same, this short-term memory is the same for control mice and for mice that have been maintained with these cycles of rejuvenation. But if we do the same test 5 days later, this is long-term memory. Now, the control mice, they don't remember that they had seen this object before, and they spend the same amount of time in the new object and in the old, 50% each time. However, the ones that were rejuvenated or treated with the Yamanaka factors, they remember that this object is an old friend, and they go directly to the new object. That is the one that calls their attention. I'm going to finish saying what are we going, in which direction. We are trying to understand in vivo the rules, the barriers, and we are trying to have a pharmacological toolkit to manipulate in vivo reprogramming with the Yamanaka factors, and perhaps eventually without the Yamanaka factors. That would be our dream. We found a number of years ago that IL-6 is an important factor in the microenvironment, and by injecting recombinant IL-6 or palbociclib, we can increase in vivo reprogramming. We have work in preparation by Elena showing that the natural killer cells are a main extrinsic barrier for reprogramming. If we deplete the mice with anti-NK antibodies, reprogramming is massively boosted. And we have another work also submitted by Marta Kovacheva, finding a metabolic requirement. She found that vitamin B12 is highly demanded by this process of dedifferentiation and redifferentiation, and the mice reprogram better if we supplement the food with vitamin B12. And with this, I will finish. I'm sorry I took maybe too much time, and maybe there's time for a question. I will be happy to answer questions either now or by email. Thank you very much.
Thank you, Manuel. Great talk. So let's give a round of applause and also virtual applause. So everybody who saw this lecture, took a screenshot, please do share with the hashtag ARDD 2021. You can also maybe use that to ask questions. There is a lot of questions on Slack. So everyone who is participating virtually, please do use Slack. So here is one question from Wilbert Vermege. Combining two most famous 2013 papers, do you think that some hallmarks of aging are more easily rejuvenatable by the Yamanaka factors compared to the other hallmarks?
Yeah, we think so. We are studying that. We think that the main impact of the Yamanaka factors is on the transcriptome and in the epigenetics, in the nucleus. But we also think that secondary to that, there is a rejuvenation in the other hallmarks. For example, senescence. We know that the Yamanaka factors have a hard time in reprogramming senescent cells. So the impact on senescent cells is minimal after one cycle. They don't rejuvenate those cells. The senescent burden remains the same after one cycle. However, if we continue with more cycles, the microenvironment is going to change. The rejuvenated cells are going to improve the immune system and the immune clearance. And secondary to the first changes in the chromatin and in the transcriptome, we think that we will observe changes in the other hallmarks. So we think that true rejuvenation actually involves all the hallmarks. The Yamanaka factors have a direct impact on the reprogramming of the transcriptome and the epigenome.
So maybe just one very quick question. Thank you, because we're kind of running out of time and the next speaker is supposed to speak right now. But one question from Ronald Cutler. Since mice and their methylation at CPG sites become more variable with age, do you think that OSCAM needs to be dosed differently depending on the old methylation age to avoid the overshoot?
Yeah, we don't know. It's a very good question. And we don't know the answer for that. Yeah, it's a good question. Thank you.
Yes, and there are multiple other questions. If you can sign up for the Slack channel and maybe answer them, that would be phenomenal. It's a great platform. We currently have more than 1,000 people asking questions on this platform. So thank you very much, Manuel. And let's give another round of virtual and real applause. So deeply admire your work and looking forward to more papers and seeing your age index exceeding your age at least 2 times. I think you're already there, right? So thank you.